Hello Georges, I am Isa Rizal.
I am Masud Acharya.
So we are pitching our project that is STT mutation detection and gene regulatory network. So introduction to Huntington disease. Huntington disease is a progressive neurodegenerative genetic disorder which is caused by the mutation in STT gene with an abnormal CAG repeat expansion which is generally inherited in autosomal dominant manner. The symptoms and severity of the STT ranges from the motor symptoms, cognitive symptoms to psychiatric symptoms. The severity based on CAG repeat is that 30 to 35 to 40 repeats is considered as an intermediate or reduced penetrance, whereas greater than 40 repeats is considered as highly severe and fully penetrant. So the problem statement of our project is that the early diagnosis of Huntington disease remains challenging. There is also a limited integration of genetic mutation analysis with a regulatory network insights, and also there is a lack of the computational framework for predictive disease progression. So we have proposed a solution for this problem, which is we have developed a computational framework for STT mutation detection. We have also integrated the gene regulatory network analysis for the disease mechanism discovery, and also use a bioinformatics tools and the machine learning approaches for predictive insights. So the framework components of our project is that we have collected the genomic data and pre-processing, and STT CAG repeat detection module, gene regulatory network construction was also done, machine learning based severity prediction, pedigree analysis and visualization, clinical decision and support dashboard. So the impact of our project is that it improves the early detection and risk assessment of the Huntington disease, which supports a precise medicine and personalized treatment planning, which enhances the understanding of the disease progression mechanism, which is generally useful for the genetic counseling, and that's it.
Now I would like to show you our project demonstration. This is our initial dashboard where we can either input the CAG count numbers or we can input the DNA count itself like this, and we can try to see if there is any mutation within the DNA count. Now this is the standard DNA sequence, so it should not have any mutation, and hence we can see that there is no pathogenic expansion detected, and we can see every regulatory network, every mutation of HDD proteins are unmutated. This is the graph of our hereditary network, and this is our parse tree. If we have any anomaly or any CAG count more than 35, it will analyze and it will detect like this, where we can see now there's a pathogenic expansion detected, and it will give a risk score of 58%, which is moderate or moderate risk. And we can see the gene expression impact, and we can see the inheritance simulator. We also have something known as gene interaction, where we can see how gene interacts with each other, and we have built a gene interaction circuit where it shows the interaction between a healthy gene and how it regulates each other. This shows the interaction of mutated gene and how it interacts with other genes, and this is the combination of both layered on top of each other, where we can see the values are constantly changing. We also have something known as the protein analyzer, where we can either paste a protein sequence of the mutated gene, or we can input the mutated DNA sequence, or we can, for the purpose of simplicity, just paste the CAG repeat numbers and it will generate for us the protein. The protein generated would look something like this. It is generated using AlphaFold, and we have various ways to view this protein, and we can drag. We also have something known as the drug interaction model, where we can choose between three different types of drugs and how the drug concentration changes its interaction with the protein, and if that changes makes any difference. We also have dose response curve. We also have the binding site maps, and how the fitness landscape shifted after the after the drug intake. This is simply the drug interaction model that we made.